Eukaryotic genomes. Eukaryotic genomes are located on several chromosomes. In case of eukaryotic genomes, there is relatively low gene density. In addition to nuclear DNA, in case of eukaryotes, there is also genome which is called as organelle genomes. Organelle genomes can be in the form of mitochondrial genome or in the form of chloroplast genome. So in case of eukaryotes, we can see that there is nuclear genome which is present in the nucleus, while in addition to nuclear genome, there is also organelle genome that can be in the form of mitochondria and that can be in the form of chloroplast. Eukaryotic genome contains repetitive sequence. Most of the DNA or the genome of the eukaryotes is repetitive in nature. So repetitive DNA can be in the form of signs which are called as short interspersed elements, and it can be in the form of lines which are called as long interspersed sequences. If we try to differentiate between lines and signs, lines, they are actually 1 to 5 kb in length and their copy number can be from 10 up to 10,000. While in case of signs, the size of the signs can be 200 base pair up to 300 base pair and their copy number can be up to 100,000. Highly repetitive DNA sequence which is present in the eukaryotic genomes that can be of different types. It can be mini satellite, it can be microsatellite, and it can be present at the telomeres. Mini satellite repeats of 15 to 500 base pair. If the repetitive DNA is between 14 up to 500 base pair, it is called as mini satellite DNA. It can be from 1 up to 5 kb long and it is usually scattered throughout the genome. On the other hand, microsatellites are repeats up to 13 base pair. Microsatellites can be in the form of single nucleotide, repetition of single nucleotide, dinucleotide, trinucleotide, tetranucleotide, and so on. So if there is repetition of single nucleotide or dinucleotide, trinucleotide, then they are called as microsatellites. Repetitive sequence in case of eukaryotic genome is also present at the telomeres. In case of telomeres, there are short repeats up to 600 base pair and they are from 250 up to 1000 at the ends of the chromosome. This is the detail of the elements or the eukaryotic genome which is present in different eukaryotic genomes. We can see that in case of eukaryotic genomes, chromosomes, they can be linear. There is centromere in the middle of the chromosomes. Then there are telomeres at the ends of the chromosome. In the chromosomes, there is origin of replication and there are replicones. In case of eukaryotic genomes, there are protein coding genes and spliceosomal introns. Genes for non-coding RNAs are also present on the genomes of the eukaryotes. Those RNA coding genes, they are genes for ribosomal RNA, for transfer RNA, for snRNA, for snRNA, and for microRNAs. So genes for different types of the RNAs, they are present on the eukaryotic genomes. In addition to these genes, there are also present mobile genetic elements. they are also the part of the eukaryotic genomes. In eukaryotic genomes, there are also pseudogenes as well as processed pseudogenes. On the eukaryotic genome, there are satellite DNA. Satellite DNA can be in the form of microsatellites, it can be in the form of mini satellites, and it can be in the form of repeated sequences. So, in case of eukaryotic genome, we also see that there are fragments of organelle DNAs. This is just one of the example of the comparison of two different genomes. One of the genome is of Homo sapien and the second genome is of a yeast. In case of Homo sapien, we see that there are approximately 23,000 genes which are present on the genome of the humans, while in case of yeast, there are approximately 6,000 genes which are present on the genome of the yeast. So here is the percentage of the coding exons. In case of yeast, we see most of the genome is coding in nature, while there is a less number of introns which are marked with a red color than mobile elements and the other genes. While on the other hand, in case of eukaryotic, in case of human genome, we see that the coding DNA is very less. We can see here, while this is the representation or the percentage of the introns and pseudogenes or the UTR regions which are present in the genome of the humans, and these are the mobile elements, mean those DNA that move from one location to another location. So all other genes, they are represented by green color. So this is the comparison between two genomes. One of the genome is of a yeast, which is a eukaryote, and the other genome is of humans, which is also eukaryote. In case of humans, we see that most part of the genome is non-coding. Just 1.5 up to 2% of the genome is coding, while the remaining genome is non-coding. While in case of a yeast, most part of the genome is coding. This is the comparison between the prokaryotic genome and the eukaryotic genome. In case of prokaryotic genome, usually it is circular and small in size. It is found in the nucleoid region and less elaborately structured and folded. While on the other hand, the genome of the eukaryotes, it is complex, it is large in size, and the large amount of protein are produced to form the chromatin or the structure of the chromosomes. It is highly extended and tangled during the interphase, and it is found in the nucleus. So this is the comparison between the genome of the prokaryotes and the genome of the eukaryotes. If we conclude that, what is eukaryotic genomes? In case of eukaryotes, the genome is located in the form of chromosomes, or it is present in the form of chromosomes within the nucleus. In case of eukaryotes, there is a low gene density, and chromosomes, their size vary. Some of the chromosomes, they are large, and some of the chromosomes, they are small. In addition to nuclear genome, which is present within the nucleus, there are also organelle genome, that is in the form of mitochondria or in the form of chloroplast.